Ease into the quiet, let your shoulders soften, and let the night take the weight from you. Just for tonight, we'll drift into the deep dark waters where strange creatures softly glow. If you drift off easily here, subscribe. A new one arrives every night. In 1930, a steel sphere roughly the size of a small wardrobe was lowered by cable into the Atlantic Ocean off the coast of Bermuda, and the man pressed against its single circular porthole had no reliable way to know whether anything was looking back at him. William Beebe was a naturalist by training and disposition, someone who had spent his career recording what the world contained, which had not, until this moment, required him to descend into it in a sealed metal ball. The sphere was called a bathysphere, a name drawn from the Greek word for deep, and it had been designed by his colleague Otis Barton to go where no vessel of its kind had gone before. Its walls were 4 centimeters of cast steel. Its porthole was fused quartz, 3 centimeters thick, set into a circular frame. The interior was just large enough for 2 men to crouch in without being comfortable, which was, in any practical sense, the only interior space available at that depth. What waited outside was something Beebe had read about and speculated about and corresponded about at length, and still was not prepared to see. As the bathysphere dropped below the surface and the green Atlantic water closed overhead, the light changed. It went, first, from the pale shimmering green of shallow coastal water to something deeper and more saturated, a blue that grew richer as the cable paid out and the sphere continued down. The world through the porthole became slowly less lit. Shapes outside grew less distinct. The bright surface receded above and was replaced by a quality of light that Beebe would later describe as the most beautiful and most alien blue he had ever seen, a color with no equivalent on land, a blue that was itself a form of dimming. Then, at some depth that could not be precisely marked, but that came as a physical sensation more than a visual one, even that blue was gone. What Beebe saw next were lights, not the lights of any instrument or equipment, not bioluminescence he had brought with him. In the absolute dark of water, somewhere near 435 meters below the surface, a depth no human eye had reached, while remaining inside a living human body, there were organisms moving in the black water beyond his porthole, and some of them were producing light. He recorded them as best he could, describing shapes and colors and behaviors with the vocabulary available to a man who had never seen anything like them. Luminous shapes, drifting cold fires, things that pulsed and receded and could not be named because no one had collected them intact, studied them in lab conditions, or given them names that would have been known to him. He pressed close to the glass and reported what he saw, and when the bathysphere was winched back up to the surface, and he climbed out into the afternoon light of the Atlantic, the scientific community received his descriptions with a mixture of interest and skepticism that would persist for some time. The organisms he saw were real, the lights were real, but the world that produced them was one that human instruments had only just begun to reach, and what Beebe had witnessed through that porthole was only the outermost edge of a zone that extended far below where his cable had been able to take him. The bathypelagic zone is the portion of the ocean that lies between 1,000 and 4,000 liters below the surface. It is not a precise or legally bounded region. Its limits are defined by physical conditions rather than any administrative agreement. By the point at which the last trace of solar light disappears above it, and the point below which the permanent cold of the abyssal zone begins to differ in character from the cold that defines it. Within those limits, the zone holds an enormous volume of water, a volume far larger than any terrestrial environment on Earth, a volume that is, in the physical sense, one of the most consistent environments on the planet, varying very little in its essential properties from one location to another or from one century to the next. Pressure inside the bathypelagic zone ranges between approximately 100 and 400 atmospheres, depending on depth. 1 atmosphere is the pressure of the air column above a person standing at sea level, the weight of the sky pressing downward without being felt because the body is built to exist within it. At 100 atmospheres, that familiar unfelt pressure has been multiplied by a factor that makes the inside of the steel bathysphere seem almost fragile by comparison. At 400 atmospheres, the pressure is sufficient to compress water itself, not into something solid, but into a measurably denser liquid than the water at the surface. The organisms that live inside this zone do not experience this pressure the way a surface creature would, because they have never lived outside it. Their bodies are built within it. Their chemistry functions within it. The pressure is not a force they are resisting. It is simply the physical weight of the world at the depth where they have always been. Temperature throughout the bathypelagic zone stabilizes across almost all of its extent in a narrow range between approximately 1 degree and 4 degrees Celsius. This stability is one of the zone's most fundamental characteristics, and it is worth sitting with for a moment because of how complete it is. On the ocean surface, water temperature follows the seasons, warmer in summer, cooler in winter, variable by latitude and by storm. In coastal and shallow waters, temperature shifts with currents and with the weather systems overhead. None of this reaches the bathypelagic zone. Below 1,000 meters, the water has been isolated from seasonal variation for long enough that the concept of season has no physical meaning. Whether it is winter or summer in the world above, whether a warm year or a cold one has settled over the northern hemisphere or the southern, the temperature at 1,200 meters or 2,000 meters or 3,000 meters is approximately what it was the year before, and the year before that, and for a very long time before either of those. The cold there is not responsive. It is simply settled. The ocean did not reveal this fact easily or quickly. For a long period of history, it was assumed, reasonably, given what anyone could observe, that the deep ocean was either empty or essentially so. The pressure at depth was understood to be immense. The darkness was understood to be complete. It seemed logical to conclude that nothing could persist under those conditions, and that conclusion was held by reasonable people with considerable scientific standing for longer than might seem, in hindsight, warranted. The correction came slowly, accumulating from fragmentary evidence that was difficult to interpret and easy to explain away. Then, beginning in the 1870s, the evidence became harder to dismiss. The expedition of HMS Challenger sailed between 1872 and 1876, covering roughly 68,000 nautical miles across the Atlantic, Pacific, Indian, and Southern Oceans. The Challenger was a Royal Navy corvette that had been modified for scientific purposes, its gun emplacements replaced with laboratories, its crew supplemented with civilian scientists, its mission explicitly redirected from warfare to inquiry. Among the instruments carried aboard were dredges, weighted frames strung with nets that could be lowered on long cables to the sea floor and dragged along the bottom before being winched back up. What the dredges brought up, over and over, from depths of thousands of meters, was life. The organisms arrived on deck in poor condition. The process of hauling a net through thousands of meters of water column was not gentle, and fragile things did not survive it intact. What reached the surface was broken, compressed, desiccated by the transition through different pressure zones, often unrecognizable as the living creatures it had been minutes earlier. But enough structure remained in each hull to establish that the seafloor at extreme depth was populated, that organisms were present there, living organisms, organisms that had adapted to conditions that the scientific consensus of the time had judged incompatible with life. The Challenger data was published over the following decades in an enormous series of scientific reports and is considered the founding document of modern oceanography. What it established, in broad terms, was that the question of whether the deep ocean contained life had an answer, and the answer was yes. What the Challenger expedition could not establish, what no instrument of the time could have established, was whether the organisms living in the deep ocean produced light. The dredging process destroyed whatever bioluminescence the animals might have had. The creatures arrived on deck, either dead or dying, their soft tissues damaged, their chemical systems interrupted. Any light they had produced in the dark water below was not something that survived the journey upward, and the scientific tools available to study it did not yet exist. The observation of deep-sea bioluminescence as a living phenomenon in animals moving through their actual environment required a different kind of instrument. It required a vessel that could go where the creatures were and look at them without killing them first. Beebe's bathysphere was, in 1930, that vessel, imperfect, limited to depths its cable could reach, unable to move laterally through the water, dependent on the surface ship for every breath and every communication. But it could descend. It could hold 2 men alive at depths no human had survived before, and it had a porthole. What Beebe recorded through that porthole in the depths off Bermuda were organisms he could observe but not collect, moving in their natural habitat for the first time in documented human history. The luminous shapes he described were real animals going about their ordinary business in water where the pressure was many times what the surface would bear, and the temperature was just a few degrees above the freezing point of salt water. They were not performing for him. They were simply living in the place where they had always lived, and their lights were serving whatever purposes those lights had always served, and the fact that a man was watching through a circle of fused quartz had no bearing on any of it. The zone that Beebe glimpsed on that descent had been undisturbed in any meaningful sense for the entirety of human history before it. It was dark before the first human eye evolved that could perceive darkness. It was cold and pressurized and populated before any surface creature had developed the language to describe cold or pressure or population. The organisms drifting in it had been living there long before there was anyone to wonder about them, and they continued living there after Beebe's bathysphere was winched back to the surface and taken apart and examined and written about and argued over by people who had not been inside it. Between approximately 200 and 1,000 meters of depth, there is a layer of ocean with a different character from both the sunlit water above and the stable bathypelagic zone below. This is a region where oxygen concentrations drop markedly, where the dissolved oxygen in the water falls, in many areas below 0.5 milliliter per liter. The depletion happens because organic material falling from the surface waters above is decomposed by bacteria at these intermediate depths, and the decomposition process consumes oxygen faster than the ocean circulation can deliver fresh oxygenated water from elsewhere. What results is a band of water that is, by the standards of most large marine animals, nearly airless, inhospitable, by the conventional understanding of what most creatures require to persist. Certain organisms have made this layer their permanent home, not as an accident and not in spite of its low oxygen, but in an arrangement where the very inhospitability that excludes most predators creates a kind of stability. The oxygen minimum zone, as it is called, functions in this sense as a refuge, not warm, not comfortable by any surface standard, but reliably consistent and reliable in its exclusions, an organism that can tolerate very little dissolved oxygen gains. In that tolerance, a kind of protection that does not require any defensive anatomy. The layer protects it by being what other creatures cannot endure. The boundaries of the oxygen minimum zone vary by location. In some ocean regions, the depletion is more severe. In others, it is shallower or deeper in the water column. What it shares across its various manifestations is the quality of being a permanent feature of the ocean's vertical structure, not a temporary condition caused by pollution or unusual weather, but a stable characteristic of how the open ocean processes the rain of organic material descending from its surface. It has been there for a very long time. The organisms that live within it have had a very long time to arrive at their arrangements with it. Below the oxygen minimum zone, the bathypelagic proper begins in earnest, and from there the conditions change only slowly with increasing depth. The cold grows slightly more intense, the pressure increases. The darkness, already absolute at the top of the zone, at 1,000 meters where the last blue light disappears, remains absolute through the entire extent of the zone and into the zones below it. There is no gradient of darkness in the bathypelagic. So, there is no slightly less dark region near the top and a more intensely dark region toward the bottom. Darkness at 1,200 meters is not meaningfully different from darkness at 3,500 meters. Both are the complete absence of solar light, a condition that is as total at the upper edge of the zone as it is at its lower. Solar light disappears at approximately 1,000 meters of depth. The process of its disappearance is gradual as a physical phenomenon, but arrives at an absolute condition when it completes. Red wavelengths of sunlight are absorbed within the first few tens of meters of the water column. Orange follows soon after. Yellow, then green, then the various intermediate colors of the visible spectrum are absorbed progressively as depth increases. What persists longest is blue, a narrow band of short wavelength light that travels farther through seawater than any other color, penetrating to depths where no other wavelength has survived. In the mesopelagic zone, between roughly 200 and 1,000 meters, this last blue light grows fainter and fainter, detectable to instruments and to the eyes of organisms adapted specifically to register it. At 1,000 meters, the last of it is gone. The absorption is complete. Below that depth, there is no photon of solar origin remaining in the water column. None has reached that depth since it left the sun. The physical process of photosynthesis, the conversion of light energy into chemical energy that sustains virtually all life on the ocean surface, is not merely difficult or inefficient at 1,000 meters. It is physically impossible because the energy that drives it is no longer present. The world below that line has been dark for as long as the ocean has held water. Not dark in the way a room is dark with the lights off, where traces of light still enter under doors, and the eye adjusts to find faint shapes. Dark in the absolute sense that physics produces, when all of the relevant wavelengths have been removed from a medium that was never meant to transmit them far in the first place. Dark without any exception built into it. Dark as a permanent condition of the physical environment, rather than as an absence of something that was recently present. Life found a way into it anyway, and has been there since long before any person thought to look. The Challenger expedition's dredges brought up the first physical evidence of that life in the 1870s, Strange, soft-bodied things deposited onto the ship's deck in the South Atlantic and the Pacific and the Indian Ocean, broken and strange, and unlike anything that had been systematically described before. The scientists aboard examined what the dredges returned and cataloged what they could identify and noted what they could not, and the notes accumulated into reports that accumulated into volumes that accumulated into the founding literature of a science that had not existed before the voyage began. Oceanography, as a modern discipline, traces its origin to that expedition, to the decision to lower nets into water that was assumed to be empty, and to discover that the assumption was wrong. The knowledge built slowly from there. Each decade added instruments more capable than the last. Each improvement in cable technology allowed vessels to reach depths that had been beyond the reach of earlier expeditions. Each improvement in preservation technique allowed the organisms that did survive the ascent to be studied more carefully once they arrived at the surface. And then, eventually, it became possible to build a vessel that could take a human being down rather than send a net. And William Beebe climbed into his bathysphere off Bermuda and pressed his face against the porthole, and the organisms drifting in the dark outside it went on with what they were doing, lit from within, unperturbed. The porthole he looked through was small. The column of water above him was enormous. The sphere was suspended on a cable of finite length in an ocean whose depths extended far below where that cable could reach, and what he could see through the glass was only a narrow cone of whatever happened to drift close enough to the sphere to be visible. He saw luminous shapes. He described them. He came back up. The zone he had briefly entered continued without him. Its pressure remained between 100 and 400 atmospheres. Its temperature held in the narrow band between 1 degree and 4 degrees Celsius, unaffected by the season, unaffected by the weather moving across the surface, far overhead, unaffected by the passage of years, or the turn of decades, or the slow progression of the century that had produced the bathysphere and the two men inside it. The cold there does not shift. The dark does not lift. The water holds its character the way old stone holds its, not because anything maintains it, but because nothing disturbs it, and the conditions that produced it remain exactly what they were. In water at 1,000 meters of depth, a thermometer would register just a few degrees above the freezing point of salt water. It would register that number in January and in July. It would register it whether the surface above was storm-tossed or still, whether the surface temperature above was tropical or arctic, whether the year was a warm one or a cold one by the measurements of the world above. The reading at that depth is not responsive to any of those variables. It reflects something deeper than surface weather, a condition produced over geological time by the circulation of the ocean itself, by the slow descent of cold, dense water from polar regions sinking along the ocean floor and filling the deep basins with a chill that spreads outward through the bathypelagic zone and persists there, season after season, unchanged and unhurried, in a darkness that is its own quiet constant. Somewhere in the deep water, in a small fleshy organ no larger than a grain of rice, a molecule is about to change. The organ is dark inside. The water surrounding the creature that carries it is dark outside. There is no light arriving from above, no gradation from dim to dimmer, no memory of blue or green in the water at this depth. The darkness is complete in every direction, and it has been complete for as long as the ocean has been this deep, which is longer than any useful comparison can stretch. And yet, inside that small organ, held inside a pocket of soft tissue, pressed against the cold and the pressure, something is about to produce light. The molecule is luciferin. It sits in the presence of an enzyme called luciferase, and when the two interact with oxygen under the right conditions, the energy released does not become heat, it does not become sound, it does not dissipate into the cold water around it in any of the usual ways that chemical energy tends to vanish. Instead, it becomes a photon, a single precise unit of visible light, released cold, released silently, released with an efficiency that no candle, no filament, no burning thing above the surface has ever matched. This is the basic event. Everything else follows from it. The luciferin molecule, oxidized in the presence of luciferase, gives off one photon of light. The organism containing this reaction produces it not as a byproduct, not as waste, but as a specific output, a controlled, voluntary release of cold light into an environment where light does not otherwise exist. The reaction itself is quiet, without heat, without combustion. In the language of chemistry, it is extraordinarily clean. What makes this process worth pausing on is not its elegance, though it is elegant, it is how old it is. The chemistry of bioluminescence, luciferin and luciferase, oxidation producing light, appears across the living world in forms so varied, so distant from one another in evolutionary terms that the only explanation is that it was not inherited. It was arrived at again and again by organisms that had no common ancestor carrying the same solution. Analyses of the genetic relationships between bioluminescent species suggest that the capacity to produce light has evolved independently somewhere between 40 and 50 separate times across the animal kingdom. The number is approximate and likely to grow as more organisms are studied in detail, but even the low end of that estimate is worth holding for a moment. 40 separate occasions on which a lineage of living things facing the same problem of darkness and the same basic chemistry available to all life on Earth arrived at the same solution independently. The solution was not copied, it was not shared. It appeared in full working form in lineages as different from one another as fish and bacteria, squid and single-celled organisms, marine worms, and deep-sea jellyfish. The convergence is particularly concentrated in marine life. The ocean, and especially its darker layers, accounts for an unusually large proportion of those independent origins. This makes a kind of sense. The ocean is where most of the dark is. The bathypelagic zone, the mesopelagic zone, the long deep water column where sunlight cannot reach. These are environments in which the usual visual tools of life simply do not function, and in which a creature capable of producing its own light occupies a genuinely different position than one that cannot. The incentive, if evolution can be said to have incentives, was present across millions of years and across an enormous range of lineages, and the answer that emerged was, again and again, some version of the same cold chemistry, the same problem, the same solution, 40 times over, at minimum. Inside the photon itself, there is something worth considering. It is blue, or more precisely, it is most often blue-green, a wavelength of light typically falling between approximately 450 and 490 nanometers toward the cooler end of the visible spectrum, in the range that the eye perceives as the deep blue of open water or the pale turquoise of shallows seen from above. This is not accidental, and it is not a coincidence of chemistry alone. It is the result, over evolutionary time, of the ocean itself acting as a filter. Seawater absorbs light. Red wavelengths disappear first, within the first few meters of depth, soaked up by the water column before they can travel anywhere useful. Orange follows. Yellow. Green persists somewhat longer. But blue, the narrow band of blue-green light that peaks around 470 nanometers, travels through seawater with a persistence that none of the other colors can match. It loses less energy to absorption with each meter it passes through. It reaches farther. In a medium that eliminates all other wavelengths over distance, blue light survives. The organisms that emit bioluminescence in this range are not emitting it because the chemistry happens to produce blue as a side effect, though the baseline luciferin reaction does tend toward blue-green. They are emitting it because across the long accumulation of evolutionary time in deep water, the versions of this reaction that produced blue-green light were the ones most likely to serve any function at all. A bioluminescent flash in the red range, at depth, travels a matter of centimeters before being absorbed by the water surrounding it. A bioluminescent flash in the blue-green range travels far enough to be detected, to communicate, to be seen. The medium selects, and what it selects for is the exact wavelength that persists. A single photon of blue-green light produced by a luciferin oxidation in the cold dark travels outward from its source with almost no loss. In the surrounding water, which has filtered out everything except this precise narrow band, it carries farther than anything else that living tissue can produce. The chemical event is small. The physics of what follows is not. There is a particular structure found in many bioluminescent deep-sea organisms that holds the whole process in a state of readiness between uses. It is a protein, and it belongs to a class called photoproteins. The way a photoprotein works differs from the simple luciferin-luciferase reaction in one important detail. It does not require the oxygen to arrive separately at the moment of use. Instead, the photoprotein molecule arrives preloaded, the luciferin and the oxygen already bound together inside it, held in a stable configuration that will not release its energy until a specific biochemical signal arrives. That signal is often a calcium ion. When a calcium ion reaches the photoprotein and binds to it, the stored complex releases. The light comes out not as a slow glow, but as a flash, nearly instantaneous, sharp, and then over. The animal can control this. The flash can be triggered, it can be timed. The photoprotein sits loaded and waiting, a compressed form of the reaction held back by nothing except the absence of the signal. And when the signal comes, the whole structure releases at once. This matters practically. An organism that relies on the slower enzymatic version of the reaction, luciferase, oxidizing luciferin in real time, produces a glow that builds and fades gradually. The timing cannot be made precise. The photoprotein system, by contrast, allows the organism to produce a burst of light in essentially no time at all, to stop it when the signal stops, and to reload the system over hours by resynthesizing the complex. In an environment where a flash of light can serve as a defense, a signal, a lure, or a warning, the ability to control the timing of that flash exactly is not a minor advantage. The loaded photoprotein molecule is one of the more specific structures in all of deep-sea biology, a molecule sized to carry just enough of the right chemistry, held in a stable configuration in cold water under pressure, waiting in the dark of a small organ inside a creature that might not need it for hours or days, and then needing it immediately for exactly as long as the signal lasts. The patience of the structure is its function. Consider what the ocean looks like in terms of the organisms that carry these systems. Estimates based on observations from submersibles and ROV surveys, approaches that allowed researchers to observe animals intact and in place rather than damaged by collection, suggest that somewhere around 76% of all organisms in the deep ocean are capable of producing light. 3/4 of everything living in the midnight zone. The figure comes with appropriate uncertainty attached to it. Counting organisms across the whole of the deep ocean is not a thing anyone can do directly, and estimates of this kind are built from sample populations and extrapolated carefully. Earlier estimates, based largely on specimens collected by trawl nets that tended to destroy the delicate light organs of many animals in the process of catching them, had put the proportion considerably lower. When the observation method changed to one that did not damage the subjects, the proportion shifted upward substantially. 76% is a number that resists easy absorption. Most environments on Earth in which life is abundant are not lit from within by the organisms that inhabit them. A forest canopy is lit from outside. A tide pool is lit from outside. Even a coral reef, which teems with color and activity, produces no meaningful light of its own. It is a reflective environment, displaying the colors the sun puts into it. The deep ocean is different. Below the point where sunlight disappears entirely, in the cold and the pressure and the permanent dark, the majority of organisms are not waiting for light to arrive. They are making it. The darkness understood this way is less complete than it first appears. It is not a featureless void. It is full of small cold lights, moving slowly, flashing briefly, glowing steadily, persisting in the water column in numbers that the surface world rarely sees and cannot quite picture. The midnight zone is not dark the way an empty room is dark. It is dark the way a clear night sky is dark, which is to say, crossed continuously by small cold points of light that become visible only when the larger lights are gone. The 76% figure held next to the image of the deep water column produces a different picture of the ocean than the one that tends to come up. Not an emptiness, not a cold silence devoid of signals, but a space lit unevenly, partially in cold blue-green by most of the things living inside it. The luciferin molecule does not know any of this. It sits inside whatever organism carries it, in whatever dark corner of the ocean that organism inhabits. And when the conditions are right, when the enzyme is present, when the oxygen arrives, when the calcium ion docks against the photoprotein, it completes its reaction and releases its photon. One photon, one precise point of blue-green light, cold, efficient, gone in a fraction of a second. And then the next one. In the deep water, this process operates in organs called photophores. The word itself simply means light-bearing, which is accurate but undersells the structures it names. Photophores come in many forms depending on the organism carrying them. Some are simple pockets of luminescent cells embedded in skin, producing a diffuse glow through the tissue. Some are more complex, backed by reflective layers that direct the emitted light outward in a specific direction, fitted with lenses that focus it, covered with pigmented filters that alter its color. Some organisms carry dozens of photophores. Some carry thousands. The positioning varies, along the belly, along the flanks, behind the eyes, at the ends of modified fins, inside the mouth. In each case, the underlying chemistry is the same basic event, luciferin, lucifer, oxygen, one photon at a time, produced cold, produced without flame, released into water that has been dark since long before anything alive today existed. The photophore as a structure is the meeting point between chemistry and anatomy. The reaction would produce light on its own if the molecules were present and the conditions met, without any specialized tissue to contain it. But the tissue matters enormously. It shapes where the light goes, how bright it appears from different angles, whether it can be controlled, whether it can be aimed. The photophore takes the basic chemical fact of the luciferin reaction and turns it into something the organism can use, something positioned, directed, timed, and modulated according to the organism's circumstances. The gap between a chemical reaction producing light and an organism using that light as a tool is bridged entirely by the photophore and the neural or biochemical systems that control it. The convergent evolution story, the 40 to 50 independent origins, is worth returning to here because it changes what the photophore represents. In many of those independent lineages, the solution was not just the chemistry, it was the whole structure, the organ, the reflector, the lens, the control system. The convergence is not merely that organisms in the dark all discovered luciferin. It is that they all arrived, independently, at the same basic architectural answer to the same question: how do you take a chemical reaction that produces light and make it useful? The answer, reached 40 times or more across the history of life in water, is some version of a photophore, a contained organ, backed by a reflective surface, covered by a lens, controlled by the organism's nervous or endocrine system, positioned where the light it emits can perform a function. The details differ. The fish's photophore is not anatomically identical to the squid's, which is not identical to the crustaceans, but the functional logic, contain the light, direct it, control it, appears across all of them, independently, repeatedly, in the permanent dark. There is something about the depth of this convergence that is worth sitting with quietly. The fact of it is well established in the comparative biology of marine organisms, examined through genetic analysis and anatomical comparison across a wide range of lineages. What it reflects is not a mystery. It reflects the power of a specific environment, cold, dark, pressurized, stable, to arrive at the same solution for the same problem across an enormous span of evolutionary time. The dark ocean is old. The organisms that live in it have had a long time to find answers, and the answers that work tend to keep appearing. The blue-green photon, the loaded photoprotein, the photophore, the cold-clean reaction, producing light from nothing but chemistry. All of it arrived, in some form, 40 or more separate times. In the mesopelagic zone, which sits just above the deep midnight water, in the range where the last dim traces of sunlight are being extinguished by the water column, the density of bioluminescent organisms is particularly high. This is in part because the mesopelagic is where the boundary lies, the zone where light from above is still present in fractional form, where the relationship between an organism's own light and the ambient light around it is most variable, and where the uses of self-produced light are consequently most complex. Organisms there face conditions that change with depth across a narrow range. A creature drifting at 200 meters is in a very different light environment than one at 600 meters, even though both are below the point where photosynthesis is possible, and even though both are by most definitions in the dark. The bioluminescent organisms of this zone have to operate in both states, in the dim residual light of the upper mesopelagic, where their own glow competes with ambient blue from above, and in the deeper darkness where no ambient light exists at all. The photophore systems that serve them in both conditions must be adjustable, and in many species they are. The brightness can be modulated, the angle can be directed, the timing can be controlled. The same cold chemistry, more carefully calibrated. Below the mesopelagic, in the full midnight zone, where the water column holds no sunlight at any depth at any time of year, the bioluminescent signals operate in a context of absolute surrounding darkness. A flash of light at these depths has no competition from ambient illumination. A glow is visible against pure black in every direction. The signals that organisms produce here, in whatever form, for whatever function, travel through water that has never at this depth been illuminated by anything other than the organisms themselves. This is the environment that 76% of the creatures living in it are equipped to illuminate in small cold flashes and steady blue-green glows continuously in the dark. The researcher Edith Witter, whose work on deep-sea bioluminescence extended across many years of submersible observation and sensitive camera deployment, contributed substantially to revising the earlier, lower estimates of how many deep-sea organisms are capable of producing light. The methodological shift she and her colleagues represented, observing organisms in place, using equipment sensitive enough to record the faint light they produced, rather than collecting them in nets that damaged or destroyed their light organs in the process, changed the picture considerably. The 76% estimate emerged from this approach, not from what arrived on deck, battered and altered, but from what was seen in the water column, intact and alive. The instruments used for this kind of observation have to be sensitive enough to detect the faint cold light against the already profound darkness of the deep water. The deep ocean is not uniformly lit at any level. The bioluminescent organisms are not glowing all the time at full brightness in every direction. The light is intermittent, faint, directional, often brief. A camera sensitive enough to record it in the water column without artificial illumination that would alter the behavior of the organisms being observed requires a different approach than the tools that work in the surface world. When those tools were developed and deployed in the deep water, the count went up. What had looked like a sparsely lit environment, based on the damaged evidence brought up in trawl nets, turned out to be considerably more thoroughly luminous than the earlier picture suggested. The midnight zone was always this lit. The instruments just had not been there to see it. A particle of organic material, drifting slowly downward through the midnight zone, colonized by bioluminescent bacteria, glows faintly blue-green as it falls. The bacteria living on it are not producing light for any reason connected to the particle's journey. They are completing their own biochemistry, their own version of the same luciferin reaction in the dark water column. The light they produce is incidental to what they are doing, which is decomposing the organic material they have colonized. But the glow is real. It falls with the particle faintly through water that is otherwise dark, another small cold light among the many that make up the deep ocean's continuous dim luminescence. The particle falls, the bacteria glow, the chemistry continues. The luciferin molecule, oxidized in the presence of luciferase, releases its photon, one cold point of blue-green light in absolute darkness, precisely where no sunlight reaches, produced by a reaction that has been arriving independently in lineage after lineage across the history of life in the sea. The number of times this chemistry was found by organisms with no connection to one another is somewhere between 40 and 50. The percentage of deep-sea organisms carrying some version of this capacity is somewhere around 76. The wavelength of light most commonly produced is somewhere between 450 and 490 nanometers, the narrow blue-green band that travels farthest through the cold, dark water without being absorbed. These are the proportions of the deep ocean's luminescence, not the light of a single species, or a single evolved strategy, or a single biochemical innovation. The light of most of the things living in the dark, produced by chemistry that arrived again and again across millions of years, emitted in the wavelength that the ocean itself has selected for, controlled by structures that organs have developed to direct and time and modulate the basic cold output of one molecule meeting another in the dark. Before any individual creature is described, Before any particular glowing animal is named or placed in its context, this is what the midnight zone holds. Chemistry, convergence, blue-green light, traveling farther than any other color in cold water. Photoproteins loaded and waiting, ready to release on signal. Photophores shaped and positioned across bodies of every kind, and across 76% of all the organisms moving through the deep water column, the same basic capacity to take the chemical fact of luciferin oxidizing in the presence of luciferase, and to make from that fact a point of cold light in the dark. The deep ocean is not a place without light. It is a place where the only light that exists is made by the things that live there, in the exact wavelength the water allows to travel, by chemistry that has been rediscovered in the dark, quietly, more than 40 times. That is the proportion of it. That is the scale. 3/4 of everything living in the midnight zone capable of this, carrying in their tissues the same cold solution to the same permanent problem, each one a small blue-green light among the many others, glowing in water that has held no sunlight for as long as the midnight zone has existed, which is longer by a considerable distance than anything carrying those photons has been alive to produce them. The loaded photoprotein sits in its organ, its luciferin-oxygen complex held in stable configuration, waiting for the signal that will release it. When the signal comes, the flash is immediate. When it is over, the protein begins its slow reload, the complex rebuilding over hours in the cold, and the pressure ready again for the next time the signal arrives. In the organisms that carry photoproteins, this cycle of loading and release can repeat many times in a single night. The light is not continuous. It is prepared, triggered, spent, and prepared again. In the organisms that glow steadily rather than flash, in the bacteria, in the simple organisms whose bioluminescence is not controlled by a neural signal, but by the continuous operation of the enzymatic reaction, the light does not pulse or flash. It simply continues, dim and blue-green, for as long as the chemistry runs. A steady glow, quiet, persistent, the basic output of a reaction that does not need to be triggered because it is never turned off. Both of these, the controlled flash and the steady glow, are expressions of the same underlying event. Lucifer oxidized in the presence of luciferase, in the presence of oxygen, producing light instead of heat, in the exact wavelength the ocean allows to travel farthest. The chemistry does not change with depth. The pressure changes. The temperature is cold and stays cold. The darkness is complete and stays complete. But the luciferin reaction, wherever it operates in the water column, produces the same photon it has always produced, cold, blue-green, efficient, released without flame or friction into water that has been waiting for it in the dark for a very long time. Somewhere in the deep ocean, across the full extent of the midnight zone, and in the mesopelagic layers above it, 76% of the organisms present are carrying some version of this capacity right now. In their photophores, or in their bacterial light organs, or in the simple luminescent cells embedded in their tissues, the chemistry is either running steadily or held in readiness. The blue-green light is being made continuously, in small amounts, by most of the things living in the permanent dark. The deep ocean, understood this way, is not a darkness waiting to be illuminated. It is an illuminated darkness, lit faintly and continuously from within by the cold chemistry of organisms that found the same solution more than 40 separate times in the exact wavelength that the cold water most faithfully carries, in a proportion that covers most of what lives there. The 76% is a quiet number. It does not announce itself. It sits at the end of a long process of careful observation, instrument development, and revised estimation, and what it says about the midnight zone is simply this: most of the things living in the dark are already glowing. The Esca is about the size of a small pebble. It hangs at the tip of a slender, flexible stalk that extends from just above the anglerfish's upper jaw, and in the cold, pressurized dark, at several hundred meters below the ocean surface, it glows, not brightly, not like a lamp or a flame. It glows the way something glows when the light inside it is very small and the surrounding darkness is absolute, a point of pale blue-white luminescence, swaying very gently with the slight movements of the water, visible from some distance away because there is nothing else to see. The light does not belong to the fish. That is the first thing to understand about the anglerfish and its glow, and it is a detail that changes the whole quality of the image once it settles in. Inside the fleshy tissue of that small hanging bulb, in structures called crypts embedded in the Esca itself, live colonies of bioluminescent bacteria of the genus Photobacterium. They are the ones producing the light. The anglerfish carries them, houses them, provides them with the warm enclosed tissue they need, and in return the bacteria glow, and the fish uses that glow as its primary tool for finding food, in a place where finding anything at all would otherwise require either luck or patience beyond measuring. The relationship is not accidental. The crypts are specialized structures, shaped specifically to hold and sustain a bacterial population. The anglerfish manages the colony housed within the Esca with a degree of control that researchers are still working to understand fully, maintaining the bacteria in conditions that keep them luminescent and viable through a life lived at pressure and temperature that would make most biological systems fail. What results is something that is neither fish alone nor bacteria alone, but a working partnership conducted in the cold and the dark, at depths where sunlight has never reached. The deep-sea anglerfish belongs to the order Lophiiformes, a group that contains dozens of species distributed across the world's oceans at various depths, with the fully bioluminescent deep-sea varieties occupying bathypelagic and mesopelagic zones, where the Esca's glow has genuine utility. It is the females that carry the Elysium, and the Esca, the elongated first spine of the dorsal fin that has been modified over evolutionary time into what is effectively a fishing rod mounted on the head of a fish. The males of many deep-sea anglerfishes are small and follow a different path entirely, but it is the female with her glowing lure that most fully represents what this group of animals has made of the dark. The anglerfish does not move quickly to hunt. It hangs in the water column or rests near the bottom, and it waits. The Esca sways. The bacteria produce their photons. In the surrounding darkness, where nothing else emits any light at all, that small steady glow is an event, a signal that draws attention, a point of interest in a world that otherwise contains none. What approaches the glow approaches the fish. The anglerfish's mouth is large relative to its body, set with long inward curving teeth, and it is positioned directly below the swaying light. The sequence of events that follows is simple and functional and has been working in the deep sea for a very long time. The bacteria inside the Esca continue glowing, whether or not anything is approaching. The light is not conditional on the fish's intent or attention. It is continuous, maintained by the bacterial colony, cycling through its ordinary chemistry, producing photons as a byproduct of a life lived inside a small tissue structure inside a fish drifting in the dark water of the midnight zone. Some distance away from the anglerfish, in the cold mesopelagic waters of the western Pacific and off the coasts of Japan, lives a creature that is less than a thumb's length long and carries not one or two light sources, but several thousand. Watasania scintillans, known in Japan as the firefly squid, produces its bioluminescent displays from a dense array of individual photophores distributed across nearly every surface of its small body. These are not bacterial light organs. The firefly squid produces its own light through a chemical process located within the photophore cells themselves, and the arrangement of those cells across the body is precise and non-random, each photophore occupying a specific position whose function forms part of a larger system. The tips of the tentacles carry the largest and brightest individual photophores, conspicuous points of intense blue light. The ventral surface, the underside of the mantle, and the lower body is densely covered with smaller light organs, so many and so close together that the squid's belly appears to glow as a continuous surface rather than as a collection of individual points. The sides and upper body carry additional photophores distributed in patterns that vary by location and apparent purpose. In total, the light organ system of Watasania scintillans is one of the most elaborate arrangements of bioluminescent structures found in any organism of its size, anywhere in the ocean. The firefly squid reaches peak abundance in Toyama Bay on Japan's Sea of Japan coast, where each spring vast numbers of individuals migrate to shallow nearshore waters to spawn. In the dark before dawn, when the squid are near enough to the surface that their collective bioluminescent displays become visible from the shore and from boats, the bay takes on an intermittent blue luminescence that has been observed and described by people living on that coast for a long time. The individual squid generating the display is small enough to sit in a cupped hand. The aggregate effect of many thousands of them moving and pulsing in the same body of water at the same time is something else entirely, a diffuse and shifting light that has no single source and no clear boundary. In deeper water, away from the spawning aggregations, Watasania scintillans moves through the mesopelagic zone as an individual animal going about the ordinary business of a squid's life, hunting small fish and crustaceans, avoiding being eaten, finding others of its species in the dark. The photophores on its ventral surface produce light at intensities that appear calibrated to the faint downwelling illumination from above, matching the background so that the squid's silhouette, seen from below by a predator looking upward, disappears against the dim ambient glow. The photophores on the tentacle tips serve a different purpose, one that researchers have described as likely involved in communication and in the recognition of other individuals of the same species in the darkness of the water column. What is notable about the firefly squid's light system is its apparent flexibility. The same animal uses its bioluminescence for camouflage and for signaling and possibly for coordination, switching between functions or running them simultaneously depending on what the situation requires. A creature 6 centimeters long carries a light-producing system complex enough to serve multiple distinct purposes at once, managed by the squid's nervous system in ways that remain only partially understood. Deeper in the water column, in a zone that presents particular challenges for any air-breathing animal trying to study it, lives a creature that occupies a taxonomic position unlike anything else in the ocean. Vampiroteuthis infernalis, a name that translates roughly as the vampire squid from hell, was given its common description by early naturalists working from preserved specimens hauled up in nets, who noted its dark coloration, the webbing between its arms, and the small spine-like projections along the inner surfaces of that webbing that looked, to eyes accustomed to surface creatures, somewhat dramatic, The name has persisted in the popular literature ever since, though the animal itself is considerably more restrained than the name suggests. Vampiroteuthis infernalis occupies a taxonomic order of its own, Vampiromorphidae, separate from the true squids and from the octopuses, a lineage that appears to have branched away from both groups long ago and has been following its own evolutionary path through the deep ocean for a time that geological evidence suggests is very long. Its closest living relatives are not among the familiar cephalopods, but in a group that includes only this one known living species. It is, in that sense, genuinely singular, not a squid that resembles a vampire, but a creature that resembles nothing else alive. Its preferred habitat is the oxygen minimum zone, that layer of deep ocean water sitting roughly between 600 and 900 meters depth, where dissolved oxygen is scarcer than at most depths, and where most large animals cannot linger for long. Vampiroteuthis infernalis has physiological adaptations that allow it to function where other cephalopods could not sustain themselves. It is a slow-moving animal, not an active predator in the way most squid are understood to be. It feeds primarily on marine snow, the organic particles, dead cells, and aggregated material that settles continuously downward through the water column from the productive surface layers above. This is an unusual diet for a cephalopod, and one suited to the oxygen-poor, energy-limited environment it calls home. When threatened, the animal's defensive response draws on its bioluminescent capacity in a way that has no parallel among other living cephalopods. Most squid and octopuses escape predators through a combination of speed and an ink cloud that obscures the predator's view, allowing the cephalopod to flee in the confusion. Vampiroteuthis infernalis, which lacks a conventional ink sac of the kind found in most cephalopods, does something different. It ejects a cloud of sticky bioluminescent mucus from the tips of two slender arm-like appendages, producing a glowing, clinging, persistent mass in the water where the animal was a moment before, and then moves quietly away into the dark. The mucus cloud continues to glow after the animal has departed. It drifts in the water, emitting light, attracting whatever attention there is to be attracted in the surrounding darkness, while Vampiroteuthis infernalis, now unlit and moving slowly, puts distance between itself and whatever prompted the response. The mechanics of this are straightforward enough once described, but the image itself is worth holding for a moment, a cloud of cold light drifting in the dark, emitting its quiet glow from a substance that was inside a living animal seconds ago and is now drifting free, the animal itself already invisible in the surrounding black. The photophores of Vampiroteuthis infernalis are distributed across its body in arrangements that include both tiny scattered light organs and concentrations of larger photophores near the eyes and along the mantle. The animal can produce a range of bioluminescent effects, from the ejected mucus cloud to pulses and glows along its own surface, and it appears capable of modulating these displays with some degree of control. In the oxygen minimum zone, in cold water barely reaching a few degrees above freezing, in near total darkness broken only by whatever bioluminescence the surrounding organisms produce, this ancient separate lineage of cephalopod has been living on marine snow and drifting light mucus for an indeterminate and very long time. In 2006, researchers operating a remotely piloted underwater vehicle during surveys conducted by the Monterey Bay Aquarium Research Institute documented a colonial organism at depths between 1,600 and 2,300 meters that was, at the time of its characterization, doing something no confirmed observation of bioluminescence had previously recorded. The organism was a siphonophore, Not a single animal in the conventional sense, but a colonial organism, a collection of specialized individual zooids organized into a coordinated structure that functions as a single entity. Siphonophores are not uncommon in the deep ocean, and their bioluminescent capacities have been known and studied for some time. This genus, Arena, distinguished itself by what color its bioluminescence was, and what that color appeared to be for. The light produced by Arena's tentilla, the small lure-like appendages that trail from the colony and are used to attract and capture prey, was red, not blue-green, not the wavelength that most deep-sea bioluminescence occupies, and not the wavelength that travels most efficiently through seawater, and not the wavelength that most deep-sea organisms can detect. Red. The tentilla were shaped and arranged to resemble small crustaceans, the kind of organisms that would constitute attractive prey for the types of fish Arena appears to be targeting, and they moved in the current in ways that enhanced the resemblance. The combination of red bioluminescent light with a morphology and movement pattern that mimicked a small animal made Arena's tentilla a prey lure in both form and spectral frequency. The significance of this was in the selectivity it implied. Most deep-sea organisms, the great majority of animals living in the midnight zone, cannot detect red light. Their visual systems are adapted to the blue and blue-green wavelengths that bioluminescence typically produces, and that faint downwelling sunlight still carries at mesopelagic depths. A red lure glowing in the deep water is invisible to most of them, but certain fish, particular species, whose visual range extends further toward the red end of the spectrum, can see it. Arena appeared to be producing a signal specifically and narrowly targeted at the subset of the surrounding community able to perceive it, using a color of light that functions in the deep ocean almost like a private frequency. The sycamore colony itself is a long, delicate structure, the kind of organism that trawl nets destroy on collection, which is part of why Arena and many of its relatives remained undescribed for so long, despite the deep ocean having been a subject of scientific inquiry for well over a century. Submersible observation changed what was findable, revealing organisms in intact living condition at depth, and making possible the kind of behavioral documentation that preserved specimens cannot provide. The red bioluminescence of Arena's tentilla was a finding that required seeing the living animal in situ, lures extended and glowing at the depth where it actually operates. Each tentillum is small. The whole colony, extending through the water column as a thin trailing structure, is far larger than the individual lures it deploys, but the lures are where the functional biology concentrates. They glow red at a depth where red light would otherwise not exist, advertising their presence in a wavelength that most animals nearby cannot perceive, waiting for the specific visitor whose eyes can register the color and whose instincts identify it as food. What the colonial organism makes of its own bioluminescence, whether there is any experience in any meaningful sense at the level of the colony or the individual zooid, is not a question with an available answer. The mechanics of the lure are observable. The fishing strategy is demonstrable from submersible footage. What persists after the description is the image of a colonial organism trailing red light at nearly 2,000 meters of depth in water that has been dark for longer than the particular lineage of humans doing the observing has existed, using a color of light as a tool in a way that took until the 21st century to find and record. In the same zone of the ocean, at similar depths, a genus of fish called Aristostomius moves through the water in a private brightness that the surrounding community cannot see. The dragonfish are a group of deep-sea predators, notable for a number of anatomical features, including teeth proportionally long relative to jaw size, a body that is slender and dark, and a set of light-producing organs positioned beneath and behind each eye. In most dragonfish, these suborbital photophores produce light in the blue-green wavelengths standard for deep-sea bioluminescence. In Aristostomius, the light produced is far red, above 700 nanometers, a portion of the spectrum that lies at and beyond the edge of what the typical deep-sea visual system can register. The dragonfish does not only produce this light, it can also see it. Research into the retinal biology of Aristostomias revealed the presence of a sensitizer molecule derived from chlorophyll that is not found in the retinas of most other deep-sea fish. This molecule shifts the visual sensitivity of the retina into the far red range, allowing the fish to detect wavelengths that would be invisible to eyes lacking it. The combination of an eye capable of seeing far red light and a photophore capable of producing it gives Aristostomias something that few other organisms in the midnight zone possess: a searchlight that illuminates only for the animal using it. The prey species that Aristostomias hunts in the midnight zone largely lack the retinal adaptation that would allow them to detect far red light. When the dragonfish moves through the water with its suborbital photophores active, the beam it projects falls across fish and crustaceans that are, in the relevant sense, not in darkness from the dragonfish's perspective, but are entirely unaware of being lit. They cannot see the light falling on them. The dragonfish can see them clearly in that light, outlined against the dark water, in wavelengths that its private visual system receives as information and that the surrounding ocean does not register at all. The hunt conducted in this private red illumination is, from the prey's perspective, happening in complete darkness. Nothing is announcing the dragonfish's approach. No photophore flicker, no movement against a lit background, no silhouette in a glow that anything nearby can see. The predator moves through its own selective spectrum, reading the water ahead of it in frequencies the surrounding community cannot access. Whether the dragonfish experiences this as hunting in light, whether there is any experiential quality to the information arriving from those chlorophyll-sensitized retinas, is entirely unknown. The physiology is documented. The experience is not available to inquiry. Aristostomius is not a large fish. In the scale of deep-sea predators, it occupies a middle position, itself a prey item for larger animals, while hunting the smaller organisms it is capable of catching. Its far-red bioluminescence and the retinal adaptation that lets it use that light are not the whole of what the fish is. They are one system among several, one aspect of a body shaped by a long history of living in cold, pressurized, dark water without any of the advantages that sunlight provides to shallower communities. But the system is a remarkably complete one. The light organ and the eye that can see it form a closed loop, a sensory and projective apparatus that works at a frequency the rest of the deep sea is not part of. The dragonfish moves, and ahead of it the water is lit in far red. To the fish drifting in that zone, the darkness is, in a specific and narrow technical sense, not absolute. It is illuminated. The illumination is private and wavelength specific, detectable only to the animal producing it, and any others that share its uncommon retinal chemistry. But it is real. Photons moving through cold seawater, bouncing off organisms that have no knowledge of being in a beam of light at all. These 5 creatures, the anglerfish with its bacterial esca, the firefly squid with its thousands of coordinated photophores, the ancient cephalopod drifting in oxygen-poor water and shedding glowing mucus when threatened, the colonial siphonophore trailing red lures for an invisible targeted audience, and the dragonfish moving through its own private illumination, share one environment and one broad phenomenon. Each of them makes light in the deep ocean. Each of them makes it differently, with different chemistry or different symbionts from different tissues at different wavelengths for different purposes. The midnight zone, understood only as a dark place where unusual things survive, contains within it a diversity of light-producing strategies as varied as the organisms that produce them. The anglerfish's esca sways in the cold water at several hundred meters of depth, a small steady glow carried by bacteria in borrowed tissue. What approaches the light approaches the fish, and the fish does not need to move to make the encounter happen. The bacteria produce their photons continuously, and the fish waits in the dark beneath the light it did not make. The firefly squid, far smaller and far faster, manages thousands of individual light organs across a body the size of a thumb, running counter-illumination and communication displays through the same system, adjusting the output of individual photophores as conditions change. In Toyama Bay in spring, several thousand of them in the same dark water produce a collective effect that is visible from the surface, an emergent glow that no individual squid generates alone. In the open water column the rest of the year, each individual moves through the mesopelagic dark, with its belly adjusted to match the ambient light above and its tentacle tips holding their brighter signals. Vampiroteuthis infernalis drifts slowly in the oxygen minimum zone, collecting marine snow, carrying its bioluminescent capacity as a defensive tool held in reserve. When the glowing mucus cloud is released, it does not resemble any defense strategy used by its distant cephalopod relatives. The cloud persists in the water after the animal has moved away, emitting light from suspended glowing particles, while the animal responsible drifts in the dark, still and unlit, at a distance that increases with each quiet motion of its 8-webbed arms. The Irena colony hangs in the water column at nearly 2,000 meters, its tentilla extended and red, a signal broadcast in a color that the deep ocean does not naturally contain, and that most of the organisms surrounding the colony cannot perceive. The lures move, the light persists. The colony does not move toward its prey, but waits for the prey, if it is the right kind of prey, to move toward the light. And Aristostomius carries the whole apparatus with it, the far-red photophores and the chlorophyll-sensitized retinas, forming a self-contained system that makes the fish's hunting ground both lit and private simultaneously. The darkness the surrounding organisms experience is not the darkness the dragonfish moves through, in the same water at the same depth, in what would register to any standard eye as absolute blackness. The fish with the unusual retina sees a world in red. The midnight zone holds all of this. It holds it in cold and pressure, in permanent darkness broken only by light that living things make themselves, in water that remains near freezing, regardless of what the seasons are doing, several kilometers above on the ocean's surface. The creatures moving through it carry their light with them, each one a brief mobile source, in a darkness that is otherwise complete, each one glowing for reasons that serve the specific life that animal is living, reasons shaped over long time by the particular pressures and constraints and opportunities of an environment that has been dark far longer than any of these species have existed. The dragonfish moves forward through the water, its suborbital photophores emitting their far-red light into the surrounding dark, the beam falling across organisms that have no way to know they are illuminated. Ahead of it, in wavelengths invisible to almost everything else in the midnight zone, the water is briefly and privately bright. A small silver fish hangs in the dark water at around 400 meters, its body no longer than a human hand, and along the length of its belly, a row of tiny light organs glows at precisely the intensity of the faint blue light filtering down from above. The match is not approximate, it is calibrated. The fish is reading the light falling on its back and producing from below an equal glow, erasing the shadow its own body would otherwise cast downward into deeper water where other things are watching. The fish is not lit up for any outward purpose a casual observer might recognize. It is making itself invisible by becoming the light around it. This process has a name, counter-illumination, though the name is less interesting than what it describes, which is a form of active camouflage, operating not with color or texture, but with light itself. Lanternfish, hatchetfish, and certain squid in the middle layers of the ocean all produce bioluminescent light from photophores arranged along their ventral surfaces, their undersides, and what they are doing is canceling their own silhouettes in real time. The physics of it are straightforward. At depth, the only ambient light comes from above, the last faint residue of sunlight, filtered through hundreds of meters of water until it is barely anything at all, a dim and directionless blue. Anything hanging in that water column with a solid body will cast a shadow downward, a dark shape against a slightly less dark background. And in a place where almost nothing is visible, a dark shape is exactly the kind of thing that draws attention. The photophores on a hatchetfish are not simply switched on and left to run. They adjust as the fish moves deeper where less light reaches from above. The organs dim as the fish moves shallower where more faint light is present. They brighten. The calibration happens within seconds, tracking changes in ambient light with the same kind of ongoing sensitivity. A thermostat tracks temperature, not a fixed setting, but a continuous response. A hatchetfish at exactly the depth where its ventral light matches the background light is effectively not there, at least from below, a small animal that has made itself into a continuation of the water around it. The family of fish called Myctophidae, the lanternfish, perform this same calibration and have done so successfully for long enough to become one of the most numerous groups of vertebrates in the ocean. Estimates of total lanternfish biomass are considerable. Some researchers have placed the figure in the hundreds of millions of metric tons globally, distributed across the deep midwater zones of every major ocean basin. They are among the dominant prey items for a wide range of larger animals. Tuna, squid, seabirds, dolphins, and deep-diving whales have all been found with lanternfish as the primary stomach contents. The counter-illumination that makes a single lanternfish harder to spot from below does not make any individual one immortal, but as a strategy scaled across a species of that abundance, it functions. The ones that glow are the ones that persist. The same basic principle, using light to manage what others perceive, operates at the other end of the interaction in creatures that have no interest in being invisible. Some animals in the midnight zone produce light not to hide, but to attract, and the mechanics of this are worth sitting with for a moment. A faint glow in absolute darkness is visible from a considerable distance. The dark does not dilute the signal, it concentrates it. Any point of light in an otherwise lightless environment draws the eye, or whatever passes for an eye in the creatures down there, immediately and reliably. A glow is not subtle in the midnight zone. It is the only thing in the room. Viperfish of the genus Chaliatus carry a modified first dorsal fin ray, a single elongated spine that arches up and then forward over the head, ending in a small glowing structure. The fish hangs more or less still in the water, the lure positioned above and ahead of its face, a faint point of light in the darkness, at close range, and deep. Anything curious about that light is also close to the fish. This alone would be an efficient enough arrangement. The light draws something near, and what is near the teeth. But the system does not stop there. Along the inner surfaces of the jaws, additional photophores cast a soft internal glow into the mouth itself, the cavity visible from some distance in the surrounding dark, a faint illuminated space that a smaller animal, already drawn toward light, might investigate further. It is a multi-stage system. First the lure above, then, for anything that comes close enough, the lit interior that functions as a kind of secondary encouragement. Whether smaller prey animals are actually drawn into the mouth by the internal photophores, or whether the jaw lining serves a different function entirely, is not something that has been definitively settled. Observing viperfish feeding in natural conditions at depth is not straightforward. Much of what is known about deep-sea predation comes from the stomach contents of trawled specimens, which tells you what was eaten but not how the catching happened, and from submersible observations that provide glimpses but rarely complete behavioral sequences. The viperfish's anatomy suggests a multi-stage luminescent predatory arrangement, and that suggestion is grounded in the specific placement of the light organs relative to the teeth and the direction of the mouth opening. What the fish does with all of it, in the dark, in the full sequence of a hunt, remains partially open. Counter-illumination conceals, allure attracts. These two strategies are almost opposites in the logic of their use, and yet both rely entirely on the same physical property, the ability of a biological organism to produce cold light from a chemical reaction inside its own tissue, with no flame, no heat, no external source required. That the same capacity can serve purposes this different from each other is part of what has made bioluminescence so common across so many different lineages. A trait that solves multiple distinct problems has more chances to become useful, and usefulness is what keeps traits around. There is a third strategy, distinct from both of these, that turns the logic of attraction and camouflage on its head and uses light for something closer to a distress signal broadcast in the wrong direction. Small planktonic crustaceans, copods, ostracods, are common prey in the midwater column. They are small and soft-bodied, not well equipped for direct defense. When a predator seizes one, the crustacean may release a sudden bright flash of bioluminescent light, not light pointed at the predator, exactly, but light produced in the immediate vicinity of the attack, which illuminates both parties to anything watching from farther away. The predator, in the act of catching the small crustacean, suddenly becomes visible. A second predator, larger, which had not registered the small drama playing out in the darkness, now has a location. The attack that was meant to be quick and unremarkable has announced itself with a flash of light, and the animal that made the light has shifted the situation from a simple predation event into something more complicated. This has been called burglar alarm bioluminescence, which is not the most formal of scientific names, but captures the function precisely enough. The small organism cannot overpower what is catching it. It cannot outrun it. But what it can do in the specific conditions of the deep sea, where predators are numerous and distributed through the water column at all depths, is attract attention to the predator. The deep ocean contains enough large hungry animals at enough different depths and positions that a sudden bright flash is statistically likely to interest something somewhere nearby that could make life difficult for the intermediate predator. The small crustacean may or may not survive the original attack, but across a population of small crustaceans that carry this capacity, the predators that make the mistake of being visible while eating will encounter more complications than those that do not. Ostracods of the genus Vargula produce particularly well-documented bursts of bioluminescent light when disturbed. Their flashes are brief and bright relative to their body size, and the chemistry behind the flash is specific to their lineage. Their luciferin compound is different from the one found in most other bioluminescent organisms. During the Second World War, dried ostracods collected by Japanese forces were used as a low-level field light source by soldiers who needed to read maps without producing the kind of glow visible to the enemy at a distance. The ostracods, ground up and moistened, produced enough light for the purpose. This is not directly relevant to how they use the flash in the ocean, but it indicates how concentrated the light production is relative to the animal's size, and why a sudden burst of it in absolute underwater darkness would register clearly at some distance. The flash of a threatened ostracod or copod is immediate, an involuntary response to the physical stimulus of being seized. In at least some species, other organisms have more control over their light production, deploying it in sequences or patterns that carry something closer to information. The lanternfish family provides the clearest examples of this. The photophores along a lanternfish's body are not arranged randomly. They occur in species-specific patterns, specific positions on the head, specific groupings along the lateral line, specific distributions on the abdomen, and these patterns differ between species and, in several sexually dimorphic species, differ between males and females of the same species. The arrangement on a male lanternfish of a particular species is not the same as the arrangement on a female of the same species, and both are different from the arrangement found in related but distinct species. In the dark water column, where visual identification by shape or color is not available the way it would be in a lit environment, the photophore pattern serves as a kind of readable signature. Two lanternfish passing in the water can identify each other's species and sex from the spatial arrangement of the light they carry. The information is not stored in the brightness of the light or in any flashing sequence. It is in the geometry of the pattern itself, a fixed arrangement, legible at the right distance to eyes that have evolved to read it. An animal looking at another member of its own species sees a specific familiar constellation of light points. Looking at a different species, it sees a different arrangement. The distinction matters for reproduction, for schooling behavior, for any interaction where knowing who you are dealing with is a relevant consideration. This quiet geometry of light, fixed patterns carrying species identity through the dark, has been documented with particular detail in the myctophid family through careful comparison of trawl specimens and submersible observations. The differences in photophore arrangement between species are consistent and diagnosable, which means taxonomists have been able to use photophore distribution as a character in species identification, the same way a bird field guide might use the position of a wing stripe or the color of a bill. The light is both something the animal uses in life and something researchers use after the fact to understand what kind of animal they are looking at. There is something quiet about the way lanternfish move through the water column in diel vertical migrations, rising toward the surface at night to feed on the plankton-rich shallow waters, then descending back into the dark during the day. The migrations cover hundreds of meters, twice a day, driven by a trade-off between feeding opportunity and predation risk. In the process, the fish carry the organic material they consume at the surface, downward into the deep ocean, releasing it at depth through respiration, excretion, and the inevitable predation that takes some of them before they complete the descent. This daily movement of carbon from the surface to the deep is part of what oceanographers call the biological pump, a mechanism by which the ocean sequesters carbon from the atmosphere into the deep sea. The lanternfish's photophore patterns, their counter-illumination, their feeding migrations, all of it is part of the same animal, and the same animal is part of a process that shapes the chemistry of the ocean on a planetary scale. The small glowing fish calibrating its belly against the faint light above is not separate from that larger system. It is embedded in it. The deep-sea hatchetfish takes the counter-illumination strategy slightly further than lanternfish in one specific respect. Its body is strongly compressed laterally, extremely thin when seen from the front, which minimizes the silhouette visible from below to an almost negligible width. The photophores are not simply on the underside, but are arranged to project light downward in a specific angular distribution that more closely matches the diffuse quality of the downwelling light from above. The light from below does not just equal the intensity above, it spreads in a way that mimics the diffuse, spread-out character of filtered sunlight seen from the deep. The hatchetfish has not simply matched the brightness, it has to some degree matched the texture. How precisely the match works, and how consistently it functions against the diversity of predators that look up from the dark, is a question that remains partially open. The eyes of deep-sea predators are adapted for detecting contrast in extremely low light, and some are considerably more sensitive than others. A counter-illumination that fools one predator's visual system might not be equally effective against a different predator with different photoreceptor sensitivities. The bioluminescent camouflage is not a single lock with a single key. It is a strategy operating within a range of conditions and against a range of sensory systems, and its effectiveness varies accordingly. The glowing ink replacement used by certain squid raises a question that the dark ocean makes specific. When everything around you is already dark, what is a cloud of dark ink for? In open water at depth, ejecting an opaque cloud into water that is already opaque with darkness accomplishes almost nothing. The predator you are fleeing is not tracking you visually in normal visible light. It is tracking you through bioluminescence, through movement, through pressure waves, through chemical signals. A dark cloud of ink in a dark environment provides no screen. Several squid species in deep and mid-water zones have addressed this in a way that initially seems counterintuitive. They have replaced the conventional ink cloud with a secretion containing bioluminescent particles, producing a glowing cloud when threatened rather than a dark one. Squid of the genus Heteroteuthis produce this kind of bioluminescent secretion. When threatened, they release a cloud of luminescent material that continues to glow in the water after the squid has moved away. The cloud does not make the squid invisible. It creates a locus of light that persists in the water, a decoy that is visible, detectable, perhaps briefly interesting to whatever was pursuing the squid, while the squid itself has already moved on into the surrounding dark. The light is not covering the animal's retreat by hiding it. The light is covering the retreat by substituting for the animal, leaving something behind that takes up space in the predator's perception, while the squid vacate the premises. The decoy principle is not unique to bioluminescent ink. Various animals in different environments use distraction displays or shed body parts to buy time for escape. What makes the bioluminescent secretion particular to the deep-sea context is the reason a dark cloud fails there in the first place. It is the darkness of the environment that has made darkness useless as camouflage, and it is the same darkness that makes a glowing cloud effective as a decoy. The ocean's permanent night has, in this case, inverted the usual logic of concealment. Being bright is the cover. The cloud from a Heteroteuthis does not fade immediately. The bioluminescent particles it contains, cells or granules depending on the species, continue to emit light for some period after being released, carried slowly by whatever current exists at that depth, spreading and dimming gradually. A camera watching from a fixed position would record the cloud as a drifting, slowly fading smear of cold light in the dark water, a shape without any animal inside it, persisting for minutes after the squid is gone. There is no urgency in how it fades. It simply dims slowly, over time, the way a coal cools after a fire, not extinguished in an instant, but releasing the last of its light in a long, quiet diminishment. Returning to the hatchetfish for a moment, the counter-illumination case makes visible a principle that runs through many of the strategies encountered in the midnight zone. The environments these animals inhabit have constrained what kinds of signals are useful, what kinds of defenses work, and what kinds of communication is possible. When vision is the primary sense mediating interaction between animals, when ambient light is essentially zero and the only light available is produced biologically, everything that depends on being seen or being hidden has to be built from biological light. There is no other material to work with. The result is an environment where an enormous variety of functions, identification, courtship, concealment, predation, defense, distraction, are all served by essentially the same physical phenomenon, a cold chemical light produced in the same fundamental way across organisms as different as bacteria, fish, squid, and crustaceans. The specificity of wavelength matters here. Most deep-sea bioluminescence produces blue or blue-green light in a range that travels well through seawater and that most deep-sea visual systems are tuned to detect. But the uniformity of that wavelength creates a particular opportunity for animals that can produce something different. If almost everything around you is signaling in blue-green, and you can produce a signal in a different part of the spectrum that your prey cannot detect but you can see, then you have something close to a private searchlight, light that illuminates the environment for you without alerting anything you are looking at. A few lineages of deep-sea fish have converged on this idea, producing far-red bioluminescence and evolving the visual apparatus to use it. The dragonfish genus Aristostomius produces bioluminescent emissions with peak wavelengths above 700 nanometers, well into the far red range, beyond the sensitivity of most deep-sea animals, which are predominantly tuned to shorter, bluer wavelengths. The photophores that produce this far-red light are located in suborbital positions, beneath and behind the eyes, oriented forward. To use a searchlight effectively, you need to be able to detect what the searchlight illuminates, which means the visual system needs to be sensitive in the same wavelength range as the emitted light. In Aristostomius, the retinas contain a sensitizing molecule derived from chlorophyll that shifts visual sensitivity toward longer wavelengths, extending what the fish can see into the far red range its searchlight occupies. The system is complete in both directions. It both produces and detects the light, and it functions in a spectral range that is, for practical purposes, invisible to the other organisms in the same environment. What this means in practice is that a dragonfish can move through water it can see, illuminated by its own far red light, while everything around it is experiencing total darkness. The prey animals it encounters are not responding to the light because they cannot perceive it. They are not startled, not fleeing, not alerted in any way to the fact that they are being illuminated. From their perspective, the darkness is intact. From the dragonfish's perspective, the darkness is not dark at all. It is lit, in a color only the dragonfish can read, showing it the shapes and positions of everything within range. The elegance of this, from an ecological standpoint, is not that it is a dramatic advantage in every circumstance. The far red light does not travel as far as shorter wavelengths through water, limiting the effective range of the searchlight, but that it solves a specific problem with a specific solution. Producing light to hunt is conspicuous. The short wavelength blue-green bioluminescence used by most organisms for signaling an attraction is detectable by most other organisms in the same environment. which means using it as a hunting light would alert prey at the same time it illuminated them. The far red solution sidesteps this entirely. The light exists for the fish that makes it, and no one else it lives alongside can see it. The information it carries belongs only to the animal that produces it. There is a kind of quiet in this. A dragonfish moving through the midnight zone in its private light is moving through a world that looks completely different to it than it does to everything else nearby. Not a more dramatic world, or a more threatening one, just a different one, with a different layer of visibility available to it alone. The differences in sensory experience between animals sharing the same physical space are not unique to the deep ocean, but the deep ocean makes them particularly clear. A fish that can see in a color no other animal in the vicinity can detect is sharing physical space with those animals while inhabiting a fundamentally different perceptual world. The range of strategies in the midwater column, counter-illumination, burglar alarm flashes, lure-based attraction, species recognition photophore arrays, bioluminescent decoy clouds, private spectrum searchlights, is not a collection of separate oddities. Each strategy represents a functional response to the same environment, the same constraints, the same opportunities. The dark provides the context. The bioluminescence provides the only material available for working within that context. Every strategy is a different answer to the same underlying question, which is, in a place where light is produced only by living things, and where almost everything that can be communicated or concealed depends on light, how do you manage what others can and cannot see? What makes the deep water column particularly rich in these strategies is the density and variety of life present there. The midnight zone is not empty. The numbers are harder to determine than in accessible surface waters, but midwater biomass estimates suggest an enormous quantity of life distributed through the water column between 1,000 and 4,000 meters. Lanternfish, bristlemouth fish, siphonophores, jellyfish, squid, shrimp, copepods, and a range of other organisms interacting across multiple trophic levels. In an environment that dense with organisms, each capable of producing light in its own specific ways, the space is threaded through at any given moment with signals, decoys, lures, identity markers, and camouflage adjustments, most of them invisible to any individual observer because they belong to sensory systems other than the observer's own. A research submersible descending through the midwater column with all external lights extinguished records something of this. The sensitive cameras on such vehicles have documented what ambient bioluminescent activity looks like in undisturbed water, a continuous low-level distribution of small flashes and glows, some brief and singular, some sustained, some moving, and some stationary. The light in the submersible's camera is not the display of any single organism, but the aggregate output of the community, counter-illumination adjusted in real time by dozens of fish at once, the brief flashes of disturbed crustaceans, the slow steady glow of organisms carrying bacterial light organs, the faint streaks of bioluminescent trails left by creatures moving quickly through the water. None of these lights was produced for the submersible's camera. They were all already there, in the dark, as part of the continuous functional activity of the community. That activity has been going on in the ocean for a very long time, far longer than any submersible has been present to record it. The evolutionary depth of bioluminescence in marine lineages, combined with the antiquity of the ocean environments these strategies inhabit, means that many of these functional adaptations have been in operation in some form across spans of time that make the history of any human technology appear momentary. The lanternfish's counter-illumination, the crustacean's burglar alarm flash, the dragonfish's private spectrum, all of these are refined products of long processes, operating in an environment that has been dark, cold, and pressurized for far longer than the particular species currently using these strategies have existed. None of this needs to be dramatic to be worth noticing. The midnight zone is not performing for anyone. The light that moves through it is functional, cold, chemical, and continuous, not ornamental, not theatrical, simply the means by which an enormous number of organisms manage their relationships with each other in the absence of any other light source. The strategies differ, the purposes differ, the organisms differ enormously in size, shape, ancestry, and sensory capability. What they have in common is the dark they share and the light they make within it. Below a lanternfish calibrating its counter-illumination, below a hatchetfish spreading its photophore light, in the texture and tone of the water above, below the drifting luminescent cloud of a threatened squid, still glowing slowly in the cold. Below all of this, the depth continues, and the dark continues, and in it the same processes run that have always run down there. Organisms communicating and concealing, and hunting, and being hunted in a medium of cold light that exists nowhere else in quite the same way. The bioluminescent ink cloud of a heteroteuthis has already been left behind, drifting slowly in the deep water after the squid that made it has moved on, still emitting its faint light, carried by whatever current touches that depth, spreading slightly wider as the minutes pass, dimming in no hurry at all, a presence in the water that the squid is no longer part of, still there, still glowing, still occupying space in the dark for a little while longer, until the light goes out of it, and the water around it is, once again, simply the water, dark and cold and full of things already moving through it, without any need of observation, carrying their lights with them into the deep. Somewhere in the cold, dark interior of a living host, a colony of bacteria reaches a threshold it has been quietly approaching for hours. The individual cells have been multiplying, packing more densely into the small chamber of tissue that surrounds them, each one releasing a faint trace of chemical signal into the fluid they share. Those signals have been accumulating molecule by molecule in the narrow space, and then, at some point that has no announcement and no ceremony, the concentration crosses a line, and the entire colony begins, all at once, to glow. The moment is not visible from outside. The host creature moves through cold water in absolute darkness, carrying this small illumination sealed inside itself. But the event is real, and it is precise, and it happens because of a kind of counting that the bacteria perform without any apparatus for counting. Each cell releases a signaling molecule as it grows. Each cell also reads the concentration of those same molecules in the surrounding fluid. When the concentration rises high enough, it means the colony has grown large enough, dense enough, and the whole population switches on together. Not because any one cell decided, but because the accumulated signal told each cell simultaneously that the threshold had been reached. The system for detecting the colony's own size is the same system that triggers the light. There is no gap between sensing and responding. This behavior is called quorum sensing. The signaling molecules are called autoinducers, and the bacterium that performs it with particular elegance, in a context that connects the microbial world to the deep ocean in a long, intricate chain, is a small marine organism called Aleva brio fischeri. On its own, a single cell of Aleva brio fischeri produces no light worth measuring. Diluted in open seawater, spread thinly through a wide cold volume, it makes no visible difference to the dark around it. But concentrated, enclosed, reaching its own threshold, the colony becomes something else, a synchronized light source, one body made of many, glowing steadily and in unison because the chemistry of its own crowding told it to. The animal that has made the best known use of this arrangement is small and soft-bodied and lives in shallow coastal waters across the Pacific rather than in the deep ocean's permanent dark. The Hawaiian bobtail squid, known to researchers as Euprymnoscolopes, hatches from its egg without any bacteria at all inside the specialized light organ on its underside. That organ exists from the moment of hatching, chambered, lined with reflective tissue, ready for its residence. But the residents are absent. The newly hatched squid enters the surrounding seawater, which holds, among everything else diluted through it, free-living populations of Vibrio fischeri, and the squid's light organ begins to select for them. The process is not passive. The organ draws seawater through itself, using cilia, small hair-like structures that create a current, and the architecture of the passages inside filters for the specific bacterium with a precision that excludes most others. Mucus secreted by the organ's cells interacts with surface molecules on Vibrio fischeri in ways that encourage adhesion and entry, while other bacterial species find no purchase and pass through. Within hours of hatching, the light organ has acquired its specific partner from the open water, and the colony has begun to establish itself inside the tissue. By the time night falls and the squid emerges to feed, the colony has grown dense enough to glow. The organ produces a soft counter-illumination on the squid's ventral surface, a downward light that obscures the animal's silhouette against whatever faint light filters down from above. Each dawn, the squid does something that initially seems to work against the whole arrangement. It expels approximately 90% of the bacterial colony from its light organ, venting most of the population out into the surrounding seawater. The light organ begins the day nearly empty. Then, over the hours of daylight, while the squid rests in the sediment, the remaining bacteria multiply. They divide and divide again inside the warm, protected, nutrient-supplied space of the organ's chambers, repopulating the space at a rate the outside environment could never support. By nightfall, the colony has returned to density. The quorum-sensing threshold is crossed again. The glow resumes, and the squid rises into the dark water with its bacterial partner restored and ready, the cycle having completed itself in the span of a single day. This daily rhythm, the expulsion, the regrowth, the threshold crossed again at dusk, has been running in populations of Euprymnoscolopes for longer than any individual squid's lifetime. It is not a behavior the squid learns. It is structural, built into the relationship between the two organisms over an enormous span of time. The squid provides an enclosed, nutrient-rich space that bacteria cannot easily find elsewhere. The bacteria provide a controlled light source the squid cannot produce alone. The arrangement persists because both parties, across millions of generations, have done better with it than without it. The light that the squid carries on its belly on any given night is not the squid's. Exactly. It belongs to the bacteria. The squid carries it and uses it and tends the colony that makes it, the way a lantern keeper tends a flame. Beyond the light organs of specific hosts, Vibrio fischeri and its relatives move through the open ocean in ways that have nothing to do with any animal's body. Free-living populations drift in the water column, colonize the surfaces of organic particles, and establish themselves on the tissue of dead organisms, settling toward the seafloor. This is where the microbial role in the deep ocean's luminescence becomes something wider than symbiosis, something that operates at a scale no individual creature commands. Marine snow is the name given to the continuous fall of organic material from the upper ocean toward the deep. It is not a seasonal event, it is not concentrated in particular places. It descends all the time, in every part of the ocean, at every depth, a slow and permanent rain of particles that includes the remains of dead plankton, shed mucus, fecal pellets, aggregated organic compounds, and fragments of organisms large and small. The individual particles are mostly small, many no larger than a grain of rice, some smaller, but collectively they represent the primary mechanism by which carbon and organic energy move from the sunlit surface layers into the deep ocean. Without marine snow, the midnight zone would receive almost nothing from the world above. Bioluminescent bacteria, including species of the genus Photobacterium, closely related to Alivibrio fischeri, colonize the surfaces of these particles as they fall. The bacteria decompose the organic material, drawing energy from the breakdown of complex molecules into simpler ones. This is not fundamentally different from what any decomposer does. What is particular about these bacteria is that they produce light while they do it, a faint, steady blue bacterial glow clinging to particles of dead organic matter as they drift downward through absolute darkness, hundreds and then thousands of meters below the last trace of sunlight. The light produced this way is not for the bacteria in the way that a signal is for its intended receiver. The bacteria are not sending the light anywhere. They are not using it to attract prey, or confuse a predator, or signal to a mate. The glow is a byproduct of the biochemistry of the light-producing system, a consequence of being alive and active in this particular way. What it means for the wider ecology of the midnight zone is another matter. Small particles of glowing marine snow may attract the attention of organisms that feed on particles. A light source in a dark place draws interest. Whether the glow of marine snow genuinely functions as an attractant for grazers, or whether it simply coincides with the kind of particle that is worth eating regardless of any glow, is a question the research has not fully closed. What the particles do, regardless of any function attributed to their glow, is fall, steadily, slowly, carrying their faint bacterial light downward through water that has been dark for longer than any measurement can establish. The fish carcass that comes to rest on the floor of the deep ocean follows a different trajectory, but arrives at a similar illumination. In the hours and days after a large fish dies and sinks to the seafloor, free-living bioluminescent bacteria colonize the tissue rapidly, drawn by the chemical signals that diffuse outward from the decomposing body. The bacteria enter the tissue, establish themselves, multiply, and begin their work of decomposition. What results, in the darkness of the seafloor where no sunlight has ever reached, is a carcass that glows, softly, faintly, its outline visible in the surrounding dark as a shape made of cold bacterial light. The glow is not permanent. It persists as long as the bacteria are active and the tissue provides what they need. As the decomposition advances and the organic material is consumed, the light gradually fades. The same phenomenon has been observed in forests, in a form that has been noticed by humans for long enough to have accumulated names in many languages. The glow of bioluminescent fungi decomposing wood in the dark, foxfire, as it is sometimes called, is a distant terrestrial equivalent of the same underlying relationship. Light produced as a consequence of microbial decomposition appearing on dead organic matter in a dark place. The fungal biochemistry involved is distinct from the bacterial biochemistry at work in the deep ocean, and the organisms involved are entirely unrelated. The convergence is in the circumstance, not the chemistry. Decomposers working in the dark, in both places, sometimes glow. The deep ocean's glowing carcasses are simply a version of a phenomenon that evolution has arrived at more than once, in more than one kingdom of life, under different conditions that shared the common feature of darkness and organic material and the metabolic activity of the organisms that break it down. There is a particular quality to the image of a fish carcass resting on the seafloor in several thousand meters of water, glowing from the activity of bacteria within it. Nothing on the surface that produced that fish, the sunlit productive waters far above, is visible from the seafloor. The light reaching the carcass from above is precisely zero. The only light present is the light the bacteria themselves produce, working steadily in the cold, in the pressure, at a depth where no process of photosynthesis has ever been possible. The glow does not attract rescue. It does not signal distress. It simply is, a consequence of life doing what life does, which is convert available energy from one form into another, and in this particular case, release a small fraction of that energy as light in the process. This is, in a sense, what bioluminescence is in its widest framing. Not a single invention, not a single purpose, not a single chemistry, but a tendency, a recurring outcome of living systems working in the dark, arriving independently at the production of light through pathways that are distinct in detail but convergent in result. The midnight zone is full of this tendency's expressions. The anglerfish carries bacterial light in the swaying bulb above its mouth. The siphonophores' trailing lures glow red in water that almost nothing else can read in that wavelength. The firefly squid's thousands of photophores pulse in patterns readable only to others of its kind. The dragonfish illuminates the water ahead with far red light invisible to the organisms surrounding it. Every one of these is a different answer to the same long-standing condition, perpetual darkness, and the advantages that light, under such conditions, can provide. And then there is the line of research that connects all of this to the world above the ocean, to laboratories and microscopes, and the inner architecture of living cells. A line that begins with a small glowing jellyfish collected from coastal Pacific waters in the early 1960s and ends with one of the most widely used tools in the history of cell biology. Icoria victoria is a jellyfish found along the Pacific coast of North America, translucent and unremarkable in appearance, except for a ring of tissue around its bell that produces a blue bioluminescent glow when the animal is disturbed. A researcher named Osamu Shimomura, working at the Friday Harbor Laboratories on Puget Sound in Washington State, became interested in understanding precisely how the animal produced this light. The work required collecting Icoria victoria in large numbers, and Shimomura did, along with colleagues and assistants, collecting tens of thousands of individual jellyfish across multiple seasons. The tissue containing the light-producing cells was removed and processed, and Shimomura gradually worked out the biochemistry involved. What he found was a protein that produced light through a mechanism somewhat different from the luciferin-luciferase system found in most bioluminescent organisms. This protein, which he called equirin, could produce blue light when calcium ions were introduced. But in the course of the work, Shimomura also isolated a second protein, one that absorbed the blue light produced by equirin and re-emitted it as green. That protein was green fluorescent protein. At the time, its significance beyond understanding one jellyfish's glow was not apparent. It was a curious component of an unusual biochemical system, interesting in the way that the biochemical details of any living organism are interesting to a researcher committed to understanding them completely. Decades passed. In the 1990s, other researchers realized that the gene encoding green fluorescent protein could be extracted from Aquaria victoria, attached to the genes encoding other proteins in other organisms, and introduced into living cells. When the resulting cells were illuminated with ultraviolet or blue light, the tagged protein would glow green, making it visible under a microscope inside a living cell in real time. For the first time, researchers had a way to watch specific proteins move, accumulate, change shape, and interact inside living cells without killing the cells, to examine them. The tool spread across biology with the speed of something that fills a gap that everyone working in the field has felt but not named. Shimomura, along with Martin Chalfie and Roger Chan, who developed and extended the applications of the protein, received the Nobel Prize in Chemistry in 2008. The protein itself is now used in thousands of laboratories across many countries, in experiments examining how cells develop, how diseases progress, how drugs interact with the machinery of life at the molecular level. None of that chain of consequences was visible when Shimomura stood at the edge of Puget Sound, collecting jellyfish in the early 1960s. The curiosity that drove the work was directed entirely at understanding one glowing animal completely and precisely. The tool emerged from that understanding, the way any good understanding of a living thing tends to produce something useful, not because the usefulness was sought, but because a real system, examined honestly, tends to contain more than the examiner was looking for. The protein exists in all of its current applications because someone became curious about how a small jellyfish made cold green light. The midnight zone is not that jellyfish's habitat. Aquaria victoria lives in shallower coastal waters, not in the permanent darkness of the deep. But the same broad phenomenon that fills the midnight zone with light connects to that jellyfish and to the protein and to the instrument light glowing green inside a cell in a laboratory far from any ocean. The thread is long. The beginning of it is the same cold light chemistry that has been running in marine organisms for hundreds of millions of years, expressing itself in form after form, in lineage after lineage, arriving again and again at the same surprising result. The quorum sensing event that began this section, that quiet threshold crossed inside a bacterial colony in the light organ of a living host, is one version of a process that is running simultaneously, right now, in bacterial populations distributed across the entire ocean, not only in light organs, not only in dense enclosed spaces where quorum sensing triggers coordinated illumination, but in the open water, on the surfaces of falling particles, inside the tissues of dead organisms settling toward the floor, in shallow coastal bays, and in the deepest places where pressure is measured in hundreds of atmospheres. Bioluminescent bacteria do not reserve their chemistry for particular occasions. They produce light when the conditions for light production are met, when the population is dense enough, when the substrate is present, when the enzymatic machinery is active. The ocean provides these conditions in more places, more continuously, than any survey has fully mapped. The extent of microbial bioluminescence in the ocean as a whole is genuinely difficult to measure. Trawl nets and sampling equipment that work well for collecting water chemistry or phytoplankton are not designed to capture the faint diffuse glow of bacterial populations spread across enormous volumes of cold water. Camera systems sensitive enough to detect the light are expensive, require deployment on research submersibles, and have so far surveyed a very small fraction of the ocean's total volume. The estimates that exist suggest that the contribution of bacterial bioluminescence to the deep ocean's overall luminescent output is substantial, possibly larger than the combined contribution of all the fish and squid and crustaceans and siphonophores producing light through their own specialized organs. The midnight zone is not primarily lit by the animals that live in it. It is lit to the extent that it is lit at all by the microbial layer that runs through everything. This is a different way of thinking about the deep ocean's light than the one that begins with a specific creature, with the anglerfish's swaying lure, or the dragonfish's private red beam, or the vampire squid's glowing ink cloud. Those are the lights that are easiest to see, easiest to describe, most available to the observer looking through a submersible's window into the dark water outside. But underneath those vivid and particular lights is a broader, less visible, more continuous luminescence, the cold, steady glow of bacteria on particles, on carcasses, in hosted light organs, in the interstitial spaces of the ocean that no submersible's camera has ever pointed at. It does not announce itself. It simply persists at every depth, at every hour, regardless of whether any host carries it or any instrument measures it. The particulate falling marine snow, glowing faintly as it descends, bearing bacteria that produce light as a consequence of their metabolic activity, passes through every layer of the water column on its way to the floor. It passes through the sunlit zone, where the bacteria's light is invisible against the brightness above. It passes through the twilight zone, where the last traces of downwelling sunlight give way to the dark. It enters the midnight zone, where its faint bacterial glow is no longer competing with anything, and it continues downward, slowly, at the pace that small particles fall through cold, dense water, on the order of meters per day for the smallest aggregates, until it reaches the seafloor or is consumed by an organism along the way, its light transferring into the chemistry of whatever eats it, the energy cycling onward into the deep ocean's food web. The bacteria on the particle do not know they are falling. They do not know they are in the midnight zone. They are doing what bacteria do, which is sense their local environment, take in available nutrients, reproduce when conditions allow, and produce light when the relevant thresholds are met. The vast scale of what they collectively constitute, billions of cells on trillions of particles descending through an ocean that covers most of the planet's surface, is not something any individual bacterium represents. It is something that emerges from all of them doing the same simple things at the same time, in the same cold dark. The midnight zone holds this. It holds all the vivid and particular lives of its named and studied inhabitants, the dragonfish, the siphonophore, the anglerfish with its borrowed bacterial light, and it holds simultaneously this quieter and more diffuse layer, the bacteria working in the dark on every available surface, the marine snow descending with its cold glow, the microbial populations cycling through their daily and seasonal rhythms without reference to any clock but their own chemistry. The named creatures and the unnamed bacteria share the same cold water, the same pressure, the same absolute absence of sunlight from above. They have been sharing it for longer than any of the phyla most of them belong to have existed in recognizable form. What persists in the midnight zone, beyond any individual creature's lifetime, is the condition itself, the dark, the cold, the pressure, and the chemistry of light production running continuously through the organisms that have made the zone their home. Bioluminescence in the deep ocean is not a novelty. It is not a recent adaptation. The oldest confirmed evidence for bioluminescent organisms in the marine fossil record is contested and fragmentary, as soft-bodied light-producing organisms fossilize poorly. But the biochemical systems involved are old. The luciferin molecules used by many marine organisms are closely related to compounds involved in fundamental metabolic processes, and the enzymes that oxidize them are distributed across such a wide range of living lineages that the underlying chemistry clearly predates any individual group that now uses it. The deep ocean has been glowing in some fashion for a very long time. It is still glowing now. In the hours that have passed since this narration began, the bacteria on marine snow particles throughout the midnight zone have continued their work. The particles that were falling at the beginning are a little lower than they were. The light organs of sleeping bobtail squid on the seafloor of coastal waters are quiet, their bacterial populations at their overnight low, slowly rebuilding toward the density that nightfall will again require. In the deep water far below any coast, dragonfish move through their private red illumination, and siphonophers trail their red glowing lures through cold water, and the glowing clouds left by fleeing squid drift in the current until the particles disperse and the light fades. None of this requires an observer. None of it has ever required one.